Alright, so let's talk a little bit about GSEA, figure out what we can do with it. So we're going to review where we are in our pipeline that we have started with. What is GSEA, what it does, how does it work, and then we're going to go into the practical. We're going to take some genes, or actually take a pre-ranked list of our differentially expressed genes, and then we're going to test that list against a few gene sets and see what we find. Okay, so this is what we've done so far. It's the same slide from last time. You had some samples, you treat them differently, or have some samples with different origins. You extract their RNA, make cDNA, ligate your adapters, throw them on the sequencer, right? So that's the biology, library prep part, and then computationally, we took that data from our whatever sequencer we had, most likely Illumina. We looked at FASTQ files, we aligned them using star aligner or top hat to convert them into SAM or BAM files. We then took tools like HT-seq to generate a count table using a reference GTF file. And then we combined those count tables into one matrix of counts for samples and genes. We then looked at one of the differentially expression software that helps us do differential expression, like DE-seq, to calculate the difference between groups. So we take our samples in groups, one group versus another group, tumor versus normal, wild type versus mutant, and calculate differential expression. At which point now we're ready to look at our set of our data set and look at gene sets and pass them through this software called David or GSEA and figure out what pathways are enriched in our data set. All right? And then that should lead to the final step, which is biological insight. So I help you all the way till David GSEA. The biological insight part is all you. That's where, you know, the ball comes back to you. It's in your code, and you take it from there. So that's the RNA-seq pipeline. So we hope to use GSEA and David to get closer to biological insight. What do we have right now is a list of differentially expressed genes. All right. So this is our starting point for today's class. I pretty much assume that everybody has gotten to this point, although I know some of you are trying to get there. And if you have any issues, again, you know, I like to iterate to everybody, please come to me if you're still stuck. I feel like I can help those who are still stuck. Usually we get hundreds of differentially expressed genes that are present. Sometimes we get none. So if you get none, I don't know what that means. I mean, sometimes it could just be that you don't have enough samples, you don't have enough room. Also, it depends on the cutoff, where you put your FDR, you know, is it 0.1, 0.2? All of those adjusted p-value cutoffs make a difference. So deriving meaning from this list is not straightforward. So gene set enrichment tries to see if specific sets of genes are overrepresented in expression data of one sample group. So this paper, this is the paper that came out from, I think it's, this one is from, I don't know whose lab it is, but it was written by, so Miyamoto did this paper, and you know, so, but essentially the point is that you have this heat map of differentially expressed group, you have all these groups. Now what does it mean to see if there are existing, you know, pathways? Can you find those pathways in your differentially expressed list? Do those pathways appear to be differentially expressed as well in your data set? So that's the whole point of GSEA, is to look at existing pathways and try to find them, how they stack up against your hypothesis. And so this was the original paper for GSEA. It's an interesting read. If you have time, you know, definitely go through this paper. It talks a little more in detail about the statistics and about the algorithm in detail as to what GSEA does. We'll try and cover some of the things from this paper today in class, but I left out most of the statistical jargon just because I thought that, you know, a lot of those calculations you probably will need a little bit more digging into to understand. So I left those out, but we'll still go over the findings, whatever you have. So this, again, this is from the Broad's website. So GSEA You can think of it as a computational method or an algorithm that determines whether an a priori defined set of genes shows statistically significant or concordant differences between two biological states. So the a priori defined set of genes, this would be your input gene set, like your whatever gene set you're looking at. This could be from MSIC-DB or KEGG pathway or any other pathway. This is your a priori set of genes. Does this set of genes show a statistically significant difference between your two biological states? So what are those two biological states? So this would be your comparison between your tumor and normal samples, right? So in your data set, how does that look? Okay, so you have your molecular profile data set, you have your gene set data set, database, you run GSEA, and then you find out how those sets are enriched in your database, all right? You get a score in the end for each gene set, so you know if you were to compare different gene sets, you can find out which gene set is more expressed in your data set versus not. Okay. So far you recognize this figure, right? So this is your two classes, class A, class B, and now you have your FDR cutoff, let's say of 0.05 or just p-value of 0.05. And so you have these genes that are upregulated in class B, but downregulated in class A, and the opposite here. So what you are trying to find out is, what is the biological meaning behind these. So what you do then is you take these other gene sets that are present in public data sets, right? So these could be whatever data sets you, pathways or gene sets that you're interested, or you're like, it's the cancer gene set pathways. These are published, usually these are published genes, okay? And then you line them up in the order of whatever order you choose. You can line them up against your data set. So what does the lining up mean? It means that you look at the genes in your gene set and try to find the location of those genes in this heat map, right? So what are your first few thoughts when you look at this gene set 1? What do you think is going on? Anything, guys? I ate a lot as well at that. So somebody needs to answer. I need to take a break.
It's a lot of genes that are overexpressed in the class A group.
In gene set 1?
Oh, sorry, I was looking at 2.
Yeah, so if you look at gene set 1, it's kind of like scattered,
Yeah.
right? It's like everywhere, you don't see any particular pattern here, right? But then let's look at gene set 2, and then as you said, you're right, that's what you find is that these gene set 2 seems to be all the genes that are upregulated in class A seem to also concordantly appear in gene set 2. Not so much here in class B, the genes that are upregulated in class B. And likewise, if you look at gene set 3, you find that gene set 3 tends to be appeared enriched in class B. So just by intuition, you can kind of get this sense like, okay, something is going on in gene set 3 and my data set. And that exactly is what is the purpose of GSEA. That's what you're trying to do, is trying to quantify this particular, this is not random. This looks like there is something that these genes are having some sort of biological meaning when you look at it in terms of your class A versus class B comparison, right? So that particular enrichment is what we're trying to quantify by GSEA. Right, we're trying to put a number on that. Yes?
What is gene set? Is that just published there?
Yes, most of the times it is a published set of genes.
And responsible for a specific event? Or
Event, right. So it could be cancer genes involved in a particular pathway,
Okay.
Wnt signaling pathway. It could be genes involved in pancreatic cancer or some other, and so these genes you already know have been published to be involved. But what you're trying to find is, how do these genes appear in my hypothesis when you're comparing my group A versus my group B, okay? And you want to put a number on it. You want to put, what is the enrichment score? Are they enriched? First of all, are they enriched? If so, are they enriched enough compared to a random set of genes? So if I was to just take a random permutation of genes and then score them in this particular case. How do that random permutation compare to my gene set, which is non-random, because this is a gene set enriched in pancreatic cancer. So that comparison gives you, is what you're interested in as well, all right? And then in the end, you want to score, that way you can compare gene set 2 to gene set 3 or gene set 1 to gene set 3. So gene set 1 tends to look like, this is what a random gene set would look like, which has no enrichment in your hypothesis. Whereas gene set 3 and gene set 2 tend to be enriched in either or direction, like one is enriched in class B, the other one is enriched in class A. Okay? That's what you're trying to look for. Okay, so these are the results that you get in GSEA. Now, how do these results stack up against here? Like, can you see how this and this are actually the same thing? Sort of? No? So it takes a little adjustment in your eyes, but basically, if you were to look at everything below this line, then these bars here are the same as these arrows here. So those bars, the blue bars, they are the position of the genes. Except now, instead of looking at it vertically, this thing has been flipped 90 degrees and is now horizontal. Here is your expression. These are your samples right about here, and these are your genes now. Okay, so the whole figure, instead of being vertical, is flipped 90 degrees, and what you're seeing here is that flipped version. Here's what's happening. Okay, so you took your class A versus class B, or disease versus normal in this case, in this heat map, and now your rows are your samples, and the columns are your genes, right? And these almost, not almost, they're always ranked in the order of your differential expression comparison. They're always ordered, right? If you take an unordered list, you cannot do GSEA, right? So the first thing you want to take into account is that you want to order your genes always. And by that I mean for your comparison. You want your, once your comparison is done, you want to order your genes in the order of your differential expression comparison, no matter what you use. Then the genes on the left side are hardly expressed in the top half. The genes on the right hand side are highly expressed in the bottom, or lowly expressed on the top half. Created is a gradient of rankless, which is what I just said, between the two phenotypes. Now, that superimposed here is what you're looking at. The gradient here is the ranked list of genes. All right, for your diseased versus normal comparison. Now, the row, I already talked about this, rows represent the samples, columns represent the genes. This part of the graph tells you how much that gene is correlated with the positive or negative phenotype in this graph. And then, here are your gene sets of interest. So, what we saw vertically, now we flipped horizontally. These are now your genes of interest. And even if you just look at their positioning, you can see that there's, this one looks a little bit random, but there is still some clustering of these genes of interest in this part. All right? So what you do then is you start this calculation, the key idea of GSEA. This is the basic crux of the entire lecture. That whenever we find a gene from the gene set, the score that we're trying to measure, we're trying to calculate this enrichment score. In the end, as I was saying earlier, for your genes of interest, you want to measure and quantify that enrichment score. Does that make sense? Yeah? Everybody with me? So in order to measure that, you start by, so you were here, just your genes, added your genes of interest, and now you're starting to quantify that genes of interest. Every time you find a match, your score goes up. Every time you find a mismatch, the score goes down. Meaning, if a gene is present, it is a positive. If the next gene that you look at is not in your genes of interest, then your score goes down until you find the next match, in which case the score goes up. All right? That's the basic idea. How do you feel about this basic idea?
So is this looking just at one gene set at a time? So we're just like, we chose one pathway and now we're looking, okay.
Take one pathway, look at the genes in those pathway, lay them right next to your ranked list of genes or with their expression values. Start calculating the presence or absence is given a score. Presence positive, absence negative.
Gotcha.
And then you just start calculating the score. So yeah, question? No? Okay, so now in the end, you start getting this familiar looking curve that you see in these papers where as this goes up, keeps going up. At one point it's gonna, because your list of genes is usually either enriched on one side or the other, so your score will start to drop if they're enriched on this side, then the score will start to go down. As you can see, it starts thinning, right? This band becomes more spacious, and at that point it starts to go down, and finally it ends. Okay, so you get this, you get this sort of curve after you calculate for all your genes, this enrichment statistic, and normally the final score of this entire curve is the highest point here and the distance of that from 0. Okay? So this height at the highest point, kind of like a mountain, look at the highest point, project it down to sea level, which is 0. That's your height or the score, the enrichment score for this gene set. Alright? Okay. Now, here is an enrichment score calculation example. So, this is your ranked list. Now, usually, when you rank your list, you want to rank it by your fold change. And so you take your fold change ranked list, and then you give it a score. So where you say that if you find a hit or if you find a presence, then that score is the sum of this mod of all the fold change divided by sigma, where sigma is the sum of fold changes for genes in the gene set S. Let's say arbitrarily that the sum of all the fold changes for your gene set S that you're looking at is 100. All right? And so that becomes our denominator, and this particular positive fold change here is the fold change of the gene that you just found. Does that make sense? So this would be for one gene, and then this denominator is the sum of all the fold changes. Cool? And similarly, there has to be a negative score if you get misses, meaning if you don't find that gene from your gene set in your ranked list, then that's considered a negative score, and that is defined by -1 divided by n minus nh, where n is the number of genes in the array, so let's say, or in your case it could be the total number of genes in your data set, which could be 20,000 or 50,000, whatever your number is. And then nh is the number of genes in the gene set S. So if you only have 20 genes in your gene set, this would be 20 divided by 1,000 or 1,020, whatever genes that you're looking at, which is a total. Okay? So the first thing that comes to mind is that the negative penalty is usually a lot smaller than the positive enrichment, which is why you get a nice curve. Otherwise, you would get kind of like an opposite direction curve. Okay, so here is your continuing running sum for your enrichment score. The way it works is, let's say you get a hit, your score goes up by 0.15, total score is 0.15. And you get another hit, this time, as the fold chain decreases, obviously this number is going to decrease. So the next positive, it's still positive, you still get 0.12, it increases to 0.27. Now you have a miss, or when you encounter a miss, you get a penalty. As I pointed out earlier, the penalty is a lot less than the positive, so your score drops, but it's a small drop. 0.269 is your cumulative score, and so on and so forth. So every time you get a hit, you start adding stuff, and then every time you get a penalty, your thing starts to go down. Eventually, the thing will flip. You get a lot more. Actually, the penalty stays constant, but the fold change continues to decrease a little bit by little. which is why your slope tends to go down eventually. All right? That's because your fold chain starts to go down for the gene. Okay? So that's why in the end you get this, final enrichment score. The maximum deviation from the top point to the bottom is your final score. Okay? In your GSEA. Okay, so the maximum value deviation from 0 from your leading sample. Okay, so finally, alright, so I'm going to do a little review. Although it's the same thing. So you have given a ranked list of all the genes on a chip based on a chosen measure. So fold change or score between two phenotypes A and B. The list is ordered, your ranked gene list. Then for each gene set you find the location. So, and then now this is your data set, ranked gene set that you have from your data. Then you take the pathway or a gene list or any list that you want to find out what the enrichment score for that list is. So here's another thing, it doesn't have to be a known pathway, you can actually take your own gene set, make your own gene set too. Alright? So any set of genes, let's say you have 20 genes that you know are implicated in some paper. You're like, oh, these genes were implicated in breast cancer. Last week I read this paper. I'm going to take this list of 20 genes. I'm going to see how they stack up against my gene set, against my data set for these two conditions. All right. So then, for each gene set, find the location of each gene in the ranked list. So this is your ranked list in your genes. This is your gene set. You find its location on your data set, and then you go ahead and calculate the enrichment score using the math that I just showed you. And then the last part is you analyze the significance of this KS-type statistics by permutation testing. So we'll go over this in a second. Basically, the idea is that now that you have your score, right, you want to make sure that you test it against the hypothesis that if we were to just take, let's say, a random set of genes, right, then would that have the same score or similar score compared to our gene set, right? Meaning that your gene set really does have significance when you look at it in our data set, or any random gene set would have the same significance in our gene set, all right? And the way you do that is, one way is you can just take, let's say you have 100 genes in your gene set, and it turns out that you have a really high enrichment score for these genes in A versus B differentially expressed comparison. Now you take a set of 100 genes just randomly, and you calculate the same enrichment score. And then you do a KS statistic test. So you can do a test where you compare all these permutations and against your statistic that you have, and figure out whether your score is truly genuine, or is it just by chance that you got this high score of enrichment. Okay? And that is done for you in GSEA, and you get that statistical test value as a significant score when you get your results in GSEA. Okay? Alright? Does that make sense? Okay. So, also another thing you want to do is you want to test for multiple hypotheses if you're doing multiple gene sets and with the false discovery rate. So normally you never test against one data set. If you're looking at MSeq-DB, there are hundreds of thousands of gene sets in MSeq-DB. So when you're getting your scores, you want to, again, just like you do with normal genes when you do a differential expression, because you're comparing two things and you're doing multiple comparisons, so you want to make sure that you adjust for that multiple comparisons with your false discovery rate. Is that point clear? Somewhat? So if you're making 100 comparisons and you have a false discovery rate of 0.1, or 1%, then 1 out of 100 you are just getting by chance. Same thing here. If you're doing 100 gene sets and you're comparing them against your data set, then 1% false discovery means that you will have one out of those 100 gene sets will be significant just by random chance. So now in real GSEA, you're testing hundreds of thousands. So if you're testing 100,000 gene sets, then you are getting what, 1,000 just by chance. So you want to do the multiple hypothesis testing correction, which is actually done for you in GSEA when they give you the results. So what you want to look at is the adjusted p-value here, or adjusted statistics, not the original p-value, okay? But this only applies if you're doing multiple tests. If you're doing your own gene set, and you only have one set of genes of 20 genes, then you don't have to do this, right? Because you're not testing for multiple gene sets. Okay. So pretty much the same thing. So these are some data sets that are there on the Broad's website, and you get this finished product where, you know, this is your data set. This is your gene set. You get your enrichment score, and then you can also get your leading edge subset. So what are your genes that are there in your leading edge, right? So that's what the leading edge is. When, you know, in GSEA you often refer to this like, oh, my leading edge subset had these genes. So these are your most enriched genes from your gene set that you just tested, okay? So it's fairly straightforward. Questions so far? Yeah? Okay. All right. So let's look at the output. So it's usually you get an output which is a HTML file. All right. So this HTML file has exactly this kind of thing. If you want to go and click on this link, it says snapshot of enrichment results. You click that and you get all the figures for all your gene sets, and then you get the top gene sets. They're ordered and ranked by the most enriched gene sets. Okay. And then, you know, if you want to go further, you can get the detailed results in HTML format, and those look like that. And then you can look at individual. Each of those are hyperlinks, you know, to other. You can look at what are the genes in those gene sets, what is the enrichment score, what are the other features. Everything is there in this, and we'll go over some of this. Okay. All right. Oh yeah. So for example, let's say we went over this link for the enrichment results in HTML format, then this thing shows up. So this basically, you can click on that, and then it gives you further details about the pathway itself, and then it also, if you click this details for that pathway, or it shows you your actual figure, which is the enrichment graph. That's the one you want to put in your slide or in your presentation. Okay. And then you also have for each gene set or each pathway, this gives you that, but you also have right here the numerics. So this is your enrichment score, this is your normalized enrichment score, and then this is your FTRQ value, p-value, and then it also tells you your leading edge score, meaning how many of the genes in the leading edge were actually present as a percentage of the total genes. So like in the leading edge, let's say 60% of the genes present in your entire gene set are present in the leading edge. That's a pretty good score to have. So you want to look at those things as well. And then it gives you the rank at the maximum. And, so I would look at, you know, your normalized enrichment score and then go from there. All right, that's the first statistic that I would look at. And usually they're ranked by a normalized enrichment score in the descending order. So that's a good thing. So you can, you know, pretty much you can start from the topmost and go from there. Okay? So here are some other things. You know, each of these has been. This slide is from the Broad's website. So, but basically, apart from the enrichment score, which is here, we also want to look at the nominal p-value. So this is that is the statistical significance of the enrichment score, and so, but this is not adjusted for gene set size, and so. So again, take this with a grain of salt that this is not adjusted. Therefore, it is of very limited use in comparing different gene sets to each other. Okay. All right. So I would suggest, you know, like reading all this again. I don't want to go into detail of everything because that's just going to take too much time. But that said, look at the normalized enrichment score, and then some of the other things here like that are interesting. Like I find this leading edge score very interesting because to me that seems to be, a good marker of how your gene set is in compared to other gene sets. So for example, if your leading edge score is 80% versus some other gene set that has 20%, then automatically that becomes, oh, obviously I'm going to look at the 80% one in this case. Okay, so let's now talk about the logistics of how to download GSEA. So you go to the Broad's website, I think you have to register. Once you've registered, then you go and look at the, it gives you this option to launch and you select the memory. Usually 4 GB is enough, but that depends on how much memory you have in your laptop. If you have less memory, you can also choose versions that are lower memory, and then you can download that. And you just click launch, and it downloads this file onto your computer, which you can double click, and it opens this software for you on your machine. And then there's a lot of information is here on the main page. If you're ever stuck, I would go on this documentation link. I definitely have gone there, and it's very useful. So if you're trying to understand, definitely read this documentation. A lot of the slides have been made from the documentation. Okay. Okay, so how do we load data in GSEA? We just click load data, and then you can browse for your files, whatever files are there. You can also drag and drop your files in here. These files need to be in a particular format. If you're using, and then you click run GSEA with these parameters. You can look at the basic fields by clicking show, and then some of the other stuff you just kind of click through these drop boxes. We're going to do this in the class, in the practical as well, so I'm going to skip this just so that we get there. Now, is anybody still using microarray data or planning to use microarray data? No? Okay. Alright, so I'm going to skip this part because this is only relevant to microarray data if you're using microarray data. Since we're not using that anymore, or I don't know if you are, but just remember that in a microarray, if you're using microarray data, then you have to enter the expression file, and GSEA's platform will do its own differential expression for you. And it also has these particular formats that the file needs to be in. It's a painful, but it's doable. I mean, you just have to convert your file into these formats, and if you are ever using microarray data, you can come back here in this presentation, and I've listed what the formats need to be and how your files need to look. So for example, your expression file needs to look like this. And then further, you have this other format files that need to look like this. Again, you're not doing any of this, so you're good. This is a descriptor file. Again, you're not doing this, but this basically, remember we have those in RNA-seq. We have a file that tells us what the samples are, and what class they belong to, what factors. So this is that factor file for GSEA. But again, we're not doing differential expression. We don't need to do this because we're not doing it. Okay. Skip. Okay. This part, this is important. So for RNA-seq, we've already done the differential expression, and therefore we have our genes pre-ranked by the order in which they are of log fold change or fold change, or any other order that you think is relevant. So we usually use a pre-ranked file or a ranked file. This should end in an RNK. This is important. It will not recognize a text file. So you need to make sure that it is a .rnk. It's essentially a text file, but you need to call it a .rnk file. Okay. It has two columns. The first line will be ignored. The second line onwards, you have your gene name, and also anything that has a hash symbol in it will be ignored. It treats it as a comment. So it has your gene name and the fold change. All right. Both of those columns are required. Right. And ideally, we would want to have it ranked by the order of the second column, which is your fold chain. Right. Now in the practical, I have given R code how to go from differentially expressed genes result to a ranked list. Right. So all of that code is there. We'll go over that. Okay. Once you have your ranked file, RNK file, go ahead and it used to be under tools, but now it's actually here, so you don't have to go to tools. You can drag and drop your data in there, select your options from here. So You select your gene sets, you select your rank file, and you click run, and boom, it gives you your the GSEA is done. Okay, so you have these are some of the existing data sets or gene sets that are existing under MSigDB. So you can download or any of these data sets and look for specific gene sets, or you can just select them in GSEA. It has that option too. You can import directly MSigDB database data sets, right? But just know one thing, that there's a lot of repetition in these gene sets. So what that means is that, let's say you have Go gene sets, and then you have curated gene sets. There'll be some gene sets that are here, that are also here, or so what you want to do is, ideally, if you're testing for a particular pathway, the best thing to do is to just select a certain set of gene sets and enter those, because you then also avoid the multiple hypothesis testing problem. Because you lose out a lot of power just because there's all this, you're correcting, right? So you lose out a lot. So that's what I would recommend, but then that also means that you have to manually create your own gene sets. Does that make sense? So it's like a catch-22, but I figure once you understand this, you can try and figure this out, right? If you don't know anything, you can still go ahead and just run the basic GSEA, and that's still good. Okay? All right, so this is just a description of all of those things. And so why do people use GSEA? Well, it's agnostic to the type of gene sets and the source of annotation, operates on an ordered gene list, pretty simple, does not require a choice of gene selection threshold nor explicit definition of significant marker set, uses distribution-free, non-parametric permutation based on test procedures with increased statistical power. Basically, it's a very foolproof, easy way to run your test for your genes. Now, if you really want to get a little more in-depth, then you can run this with selected gene sets using command line, but that gets a little harder. So I usually don't go into that for this, but if you're interested, there are R packages called GSEA. There's an R package for that. You can download that in R, and that can then just basically be run right through in your R code, as opposed to taking a break, getting your rank list. It's not too inconvenient, but let's say you want to create if this as part of your pipeline, then it might be easy to add this in the R code, right? Okay. All right, so here's another software that that's called David, and David is written by a statistician as opposed to a computer scientist who wrote the previous one, GSEA, from the Broad. Right? So the fact that David was written by a statistician, sometimes the underlying algorithm is different, and it'll give you some different results, but most people here tend to use GSEA from the Broad, so feel free to use that. I use David. Sometimes I would recommend people to use it as a sanity check. So just make sure that the gene sets that you're getting enriched also appear enriched with David. Right? So that said, since I'm talking about this, I'm going to go over in a little more detail and show you how to use it. Basically, they use these things called as distinguished genes. So these genes have PhDs. That's why they're distinguished. Or they just happen to be differentially expressed. So they're basically differentially expressed at some FDR cutoff. You already know that. Some bioinformaticians recommend analyzing significant up and down genes separately. So this is something interesting that when you're using David, I also actually recommend that. I would say that just use your upregulated genes for the first part, and then use your downregulated genes. Right? It gives you much more statistical power if you use them that way, as opposed to lumping them all together when you're using David. So here are the Davids. Just like MSigDB is the data set or pathway gene set for GSEA. David uses all of these data sets to identify pathways. So you have the go biological processes, molecular function, cellular component, COG, ontology, and here are the links for each of those. So you can individually go and check out what those pathways represent, okay? And then it keeps updating from the back end, it keeps getting new gene sets from these, so you don't have to worry about that. The website always stays updated, okay? Alright. So, so what does David do? Well, essentially, conceptually it's very similar to what I just talked about in GSEA, but let's look at this example. It does a Fisher's exact test to identify significant pathways. So, let's say in this example, you have how many genes total? 10,000. Of those 10,000 genes, 1% are in the pathway S. So you have a pathway S that you are interested in. This is your gene set, right? Now, ideally, since 100 is 1% of 10,000, correct? You would expect that if there are 50 genes in your differentially expressed gene list, 1% of those should be, there are 50 genes in your differentially expressed list. One would expect on average to have 0.5 genes of a random list of 50 genes as being in pathway S, correct? Right? So 100 is to 1,000, 0.5 is to 50, right? That's what you would expect to be enriched in your pathway. Now, but in essence, if it was a random list, 0.5 is your expectation, but in this illustration there are actually 5 genes that are in the differentially expressed gene list in S. So you have these genes 5 in the differentially expressed list from the pathway S. So these are not in, 45 are not, and the rest 95 are in pathway S that are not in the differentially expressed gene list and not in pathway S. Does that make sense? So you have a matrix. Now with this matrix you do a Fisher's exact test, you get a p-value, and you are able to figure out whether the presence of these 5 genes is more than what you would expect at chance, which clearly in this case it is. Alright? Does that make sense? But now with the Fisher's exact test, you can get the p-value for this event, or the probability that by chance one would have this many 5 genes or more of, genes of a randomly chosen list of 50 genes being in the pathway S. Alright? Do you get the idea? Yes or no? Okay, so that's the main difference between this and the other one. So this uses Fisher's exact test, that uses this convoluted enrichment score thing to come up with this exact sort of result. Okay? Alright? And the same thing, they also do multiple hypothesis testing. If you're testing multiple gene sets, each gene set is then given, you do a Fisher exact test first, and then for all the probabilities that you get, you correct them for multiple hypothesis testing. Okay? So, this is your David website. The way it works is you have this functional annotation, gene function classification, and other tools. But we're interested in this one because we want to do the enrichment analysis here. So we click this link. Okay? Or you can just go start analysis, right? So you can start that, and then it gives you this options to upload it. So it's just the interface is a little clunky, but it does pretty much the same things. It's online. You select your gene list, so you paste your gene list here, or you can upload from a file, or you can upload multiple files. So let's say you have multiple gene lists. So these are your gene sets, right? Okay. So you take that list, and then you go ahead and select your identifier. Usually it's your gene ID or gene symbol, right? In our case, it's our known gene name. And then you specify that this is a list type. You can also enter your own background list if you want to, but usually we just select the list. And then you submit this, and it goes ahead and runs your GSEA. And boom. So after that, press submit. So if you gave David gene symbols rather than array probe IDs, then you will usually see a box indicating multiple corresponding species. So that's something that you need to be careful about, and what you can do then is you select OK and then you select your species. Alright, so usually it gives you this box, just read through it and be like, oh, it looks like you think these genes are present in multiple species. Which species are we talking about? So you can select the species of Homo sapiens or whatever other you're using, and then you can start your functional annotation tool or whatever annotation clustering is usually what people do, and you can start running David with that. Then a new window that appears, click on the annotation tool, and then you can look at your annotation clustering after the results. These are your this is the number of unique gene IDs David recognizes from the submitted list, and you can do your annotation clustering, which then you can look at. It gives you a false discovery rate adjusted with the Benjamin Hofbauer correction, which I was talking about after the adjustment. So you look at those, and then you can start looking at whatever you find. What is the meaning of all this? I honestly don't know. This is the part where you guys take it from here. Okay, and then you can also click on those, and you get these keywords and go terms and all of this other stuff that we can then look at dispense. These are all clickable, so you click on them to get more information, right? Along with the scores. And let's say it takes you to the EMBL for the go terms to look up what each of those represent. Okay? All right? Oh yeah, and you can also get this. This David display from a KEGG pathway. Red stars designate genes occurring in the list submitted to David. So you can look at it as part of a pathway. Where do your genes appear in that pathway with those red stars? So I thought this was a pretty cool feature you have in David to kind of identify where does my gene actually relate to in this pathway. And then you can do that here. Okay. All right. These are more examples. I'm not going to go into this. Pretty much the same thing. Oh, you can also get a heat map when you click on these little figure things. It's a 2D view. Clicking on these shows the individual genes from the submitted list are in which gene sets of the corresponding. So different representation of the same thing, something more pleasing to the eye. All right, questions? If not, we can take a quick 2-minute break, 2:55, or let's start the practical. Yeah? Okay, so all eyes here, no hands on the computers. Okay. So first things first, as always, you'll go to the website, you can go to My Courses, and everything will blank out. Okay. So this is all planned, don't worry. Then the blue screen comes and then everything goes back. Okay, good. It's working as it should. So you're going to go here, you're going to select your course, which is this guy right here. You'll only have one, so you're good. And then scroll down to the GSEA part, which should be all the way here. It's not enabled. Okay. So that would be my issue to fix. But basically, once it is available, which should be right about now, go back. Okay. So, so you'll be able to see GSEA here. Now what you're going to do is you're going to look at these, go over here, click this. There are a couple of things you'll need to download, and that includes this rank file for your analysis. I'm assuming that most of you will not have this because the R code for that is here. And this is the DDC code with the GSEA code added. So there's a couple of lines. So let's actually think about it. So once you click this link, you'll be able to download this R file. As always, it's going to have a .txt extension. If you want to open it in RStudio, I highly recommend to remove the .txt and then open it in RStudio. Okay, so I have done that already, and then this is the file. It looks kind of like this. So this was written earlier by Mark for the practical, but then I ended up, so it's the same practical file. What you want to do is you want to scroll all the way to the end, and I have added this tag for code for GSEA, right? So when you are looking at your code here, you have this res_rank, which is a new variable that I create from ordering your log2fold change of your result object, right? So this object, if you will go up, you can scroll up. The res object is created all the way after your DSEQ. So once you're done with DSEQ, you create a results object, right? So this data frame contains your log2fold change p-value for your results. Yes.
I have a question. So for the DSEQ dataset, for example, you not only have control, but maybe you have condition 1, condition 2, and 3, and then you do the RES.
So you specify the contrast.
So what's the p-value then calculated the fold change?
If you have more than one factor and you're not specifying the result that you're looking for, it's going to compare the first two,
Okay.
right? But that's the reason why I say that you have to specify like results high, results medium, results low. You have to specify your contrast, saying which two conditions do you want the results for. That's important, okay? So once you do that, and you have your results, so either you take the results or your results high, either of or any of these. So basically, I'm assuming res or results here represents one of these guys, whichever is your condition you're interested in. Okay. So when you take that, then you rank or order everything by the fold change in your result, right? Because we want to create a ranked list of our data set for the condition that we have just compared. So given that we're comparing high condition versus low, what is the result when ordered by fold change for that condition? Okay? So let's say we order that and then we call that res_rank. Cool. Then we're going to then go ahead and create another object where we basically take two things, the name of genes and their fold change, and create a new data frame like this. This is your ranked gene list. It has two columns, the list, the names of genes in your result and their log2fold change when ordered by the rank. Clear? Yeah? Okay. And I then go ahead and set my directory here, and I write that, call it ranked gene list. Okay? Is that clear? So it's only 3 or 4 lines. All right, so you add this code, and then you add that. Okay. Now, this ranked file is, first of all, this is the R file. This is your ranked file, which is the output of this. Okay? So it's going to look something like this thing. So you have your gene name and you have your fold change. Right? And this has all your genes. So it's a pretty big file, but that's expected. This has about 20,000, 21,000 rows, which is what we expect. We want this to have these many, because this is your entire data set. This is what you're going to put in as your file. Okay. All right. So once we have this, here are your guidelines for today's practical. We go to the GSEA home, which is here, and we go here to the download, and we go ahead and download our software. Okay. So you usually ask your email, login, and then you select your memory requirements. I just say 2 GB, more than enough. Launch. It says this here, discard or keep, so you keep this, and then you look at that in Finder, wherever it is, and you could double click it. Okay, so when it says this kind of thing, what do you have to do? Go to System Preferences. Come on, man. what is it called, settings? Why did they remove that?
System preferences.
I thought, that's what I said, right? There you go. Alright, something's weird. I'm just gonna try to put it in here. I'm just going to open it from my command line, but you get the idea, right? Like this is what you're going for. And then it usually tells you that you try to download this GNLP, security and privacy, right? And here it says open anyway, and so you're going to click that open anyway, right? And then this will launch Java, which will launch the software. Boom, run, boom, and so now you have your GSEA software. Okay, so the first thing you want to look at here is there's multiple steps available here, including MCDB stuff, but we're going to start by loading our data. Or even better, we actually want to run our GSEA pre-rank, which is right here. So in order to run this though, we need to first select the gene set database. So when you click this, it gives you a list of all the gene sets that are available. Or you can select your own from using this. You can load your own file of gene sets here. So I'm going to select the hallmark or let's say, which one is, anybody have a favorite? Should I just go with hallmark? Like you have a lot of gene sets, so you know, you gotta pick the ones that you like. Let's just say hallmark and you know, gene symbol.
Do KEG.
Where's KEG?
2 down from gene symbol, I think. Is it KEG?
Oh yeah, that one. KEG, gene symbol. Oh, come on. Hallmark. Gene symbol. Okay. All right. Now you want the number of permutations. So this is the, remember in order to get the false positive rate, it does the number of permutations for your random genes. So you don't want too many permutations initially. I'm just going to select it at 10. When you run it, once this runs, you can go ahead and select 1000. So initially I'm doing 10 because I don't want it to run for too long. Your rank list, this is what you need here. So where do we get the rank list? We actually have to load that. So this is where you go back to Finder and go where your rank list is. Now you can download this from the website right here that I showed you. This is your rank list. It's going to come as a TXT file and stuff, so you got to rename it to this whatever rank list, and then just go ahead and drag and drop here, right? And select load these files. So you should get this. There are no errors. Okay. All right. So now when we go back to run GSEA pre-rank, we are ready to run. We can look at your basic fields. Now here is whatever. And then you select classic here instead of weighted, right? And the reason for that is because we're using GSEA pre-ranked. This is a parameter that always has to be classic. It cannot be weighted. Okay? Weighted is the default, but that's only for when you're running not RNA-seq, but you're running microarray data and other stuff. But most of the times you'll be running RNA-seq. So make sure this statistic, enrichment statistic, is calculated the classic way. So that's the only caveat. Other than that, you can just go ahead and click run right here in the corner, bottom most, and it should start running. And pretty soon it will tell you whether it failed or ran, or sometimes it fails for some reason because you missed something or some parameter is wrong, in which case it'll, instead of showing here success, it shows error, and then you can click that and figure out what the error is. Normally it should run. Okay, so I'm going to let it run. In the meantime, let's go ahead and go to David this is this site. Last time I checked this had an error, so I had to use this site, this development site. Well, I'm going to give it another shot and try this one more time. So, say you have a gene list. In this case, I'm going to just take a couple of genes from here. It's a lot easier when you have, Excel, but I opened it here, so select one more. Okay. All right, and then these are all right here. You select your gene symbol, official gene symbol. Okay. Once you select that gene list, you can say submit list, and then it'll give you that pop-up thingy saying, hey, you got to specify what species you are. So we're actually Homo sapiens. So we're going to say use that, and then again it gives some error. So this was the problem that I was having last time. Okay, let's click this, and even after specifying the species multiple times, they still haven't corrected this error. Anyways, so you get kind of these, functional categories, gene ontology, and you get all these charts. I didn't select a really good gene list, but you get all of those. Normally these are a lot less, not all of them are present in all of these, so you get a lot better result than this. But I leave it as an exercise for you guys to go ahead and work through this. But yeah, we're like basically, you can look at this functional annotation clustering. If you find, in this case you didn't find it. This becomes a lot better if you have a much longer list, so you get a lot more meaningful results. All right, let's go back and look at our original thing here. Okay, so this one ranked, it was a success. So when you click success, it opens this in a browser and it actually shows you. So this success is a clickable link. All right, so just go ahead and click here, and it opens a new window in your browser. And pretty much everything here is your enrichment in a simple, like, okay, these many gene sets are upregulated, these many gene sets are significant at 25% cutoff. Let's look at the snapshot. Looks pretty good to me. And then you can go and look at the detailed enrichment results. So, you have your oxidative phosphorylation. You can click on that to find out what that is, what it means. And then you can also look at this, whatever, related genes, gene sets, and then you can go into the rabbit hole and, click on these details for that gene set. When basically, it's an endless thing from here on out, right? You can look at the score, you can look at your genes that gave you this, like what is the rank in that gene list, and all of those things. All right?
Vishal.
Yes.
One thing, David does not take into consideration of the rank, the number.
No.
Just the names.
Yeah. So you look at your functional annotation. So you basically give it. That's why you only give it your upregulated genes or your downregulated genes, and not the entire thing. And ideally, you would want to give it your ranked list to begin with. But I don't think the rank matters because it basically looks at your Fisher exact test between how does it compare to a random set of 5 genes versus your set of 5 genes in existing gene sets. So that's why it's different from GSEA.
But GSEA takes the
rank.
Okay.
So you can go ahead and go from here and kind of go down this and figure out what. Like, why is COX-17 rank number 21? What does this gene do? This takes you to AFI, which is a mistake, but you kind of get the idea. You can go ahead and look this gene symbol up on your website or wherever you want to look at. Do a Google search and find out what this gene does. Right? Okay, that's pretty much GSEA. You know, it's not very complex. The part that I unfortunately cannot explain to you in this is the biological insights part, which is where you have to come back to me and tell me what you find. All right, and let me know if anything interesting comes up in this gene set, because it shouldn't, because this is just random.